Share screen.
Okay, let me introduce you, Edie, Dr. Edith London.
Hold on. Don't
Okay.
you want to let me share my screen first?
No, you could get it up while I'm talking. Dr. Edith London is a distinguished professor of psychiatry and biobehavioral sciences and of the molecular and medical pharmacology at the University of California, Los Angeles, a pioneer in brain imaging to study addictions. She has performed translational studies elucidating neurochemical and circuit level abnormalities that inform evidence-based treatments, bridging psychiatry, pharmacology, and neuroscience. Her work has provided notable first mapping drug-induced euphoria and drug craving in the human brain, visualizing the cerebral distributions of actions of abused drugs, and developing probes for external brain imaging. Focusing on corticostriatal circuitry, dopamine receptor signaling, and executive functioning, her recent work falls her seminal findings of prefrontal cortical and cognitive deficits in cocaine users, and provides a potential therapeutic target for addiction treatments and a mechanism by which dopaminergic neurotransmission modulates decision making through an interactive frontal striatal system. Dr. London is a good colleague of mine. We've worked together on several projects over these years. She's been very supportive of our SART Institute, and our talk today is entitled Imaging to Understand Drug Addiction. Thank you so much, Dr. London, for joining us.
It is my pleasure to be with you. I just have a quick question before I get started. Can you see my slide?
Yes.
Awesome.
And we can hear you.
That's wonderful. So I'm going to be talking to you today about brain imaging, and fortunately, I've been there from the very beginning. I remember when people were doing CT scans before MRI scans, and I'll talk to you about some of that, and molecular imaging using nuclear medicine probes. Just to give you an idea about the different techniques that are used, you hear a lot about MRI, and MRI can be used to measure brain structure, and fMRI, or functional MRI, is also used to measure brain function. I'll be talking about these techniques, as well as positron emission tomography, which gives us a look into the dynamics of a radioactive molecule in the brain. So just to give you an idea about how recent all of this happened, MRI was discovered in the 70s, and Lauterbur and Mansfield got the Nobel Prize for discovering and developing MRI. And shortly after they got the Nobel Prize, like the next day, Glutamate took a full-page ad in the New York Times talking about how he should have been among the recipients of the Nobel Prize because he had a paper earlier than the work of Lauterbur and Mansfield describing magnetic resonance imaging. I think a full page in the New York Times was $10,000. So how does it work? There's no radioactivity, but MRI depends upon the principle that each proton or hydrogen atom in tissue is going to spin along an axis, and in the absence of a magnetic field, the spins are kind of randomly oriented. But if in a magnetic field, the axes of rotation line up and create magnetization along the axis of the scanner. Then a radio frequency pulse is applied, and it flips the axis and frequency of the proton spin. And then if the pulse is turned off, the protons relax and energy is released, and it's the signal that's received by the cells and by the coils. And so we've learned a lot about structural MRI over the years, and there were for many years papers by individual labs where small sample sizes were studied. And that's kind of out of fashion right now. And right now, everything's about big data. And so the idea is to have consortia And there's a big consortium called the Enigma Consortium, and there is one called Enigma Addiction. And there are more than 23 labs now all across the world, and thousands of cases, case control studies, addiction versus control. And, you know, when you have studies across all of these different laboratories, you have to harmonize the data, and you lose something when you kind of average across all of these laboratories, but yet you do get some general findings. And in this first paper that came out from the Enigma Addiction Consortium, basically what was found was that the biggest effects were related to the use of alcohol. And these dark areas show where there is kind of a deficit in gray matter of the brain. And this is superior cortex, this is parietal lobe, and you have cingulate cortex. You have your biggest effects related to the use of alcohol. And there are also some general findings that were observed, and those are that there are deficits in the insula, which is kind of this little island of cortex that you can see if you peel away the parietal cortex, and the orbitofrontal cortex. This is part of the frontal lobe, of course. So why does it matter? What is the insula all about? And this is where it is. If you peel away these superficial layers of cortex, you can see it. And it's kind of tucked inside the lateral sulcus. It's part of the limbic system. It has some very important connections. No brain functions in and of itself, and it receives information from the thalamus on sensory input. It integrates data from the amygdala, giving salience to an observation, linking it to emotional memories. It's linked to the anterior and prefrontal cortices that are involved in attention, decision-making, and impulse control, and is connected to the striatum. So it kind of has a seat that connects it to data coming in and decisions going out. And it's important in evaluating choices that an individual makes. So it's activated by a variety of feeling states: pain, love, emotion, drug craving, feeling good. It's important in self-awareness, body representation, and, as I said, decision-making, including those that involve risk. There's a very famous study that was done in 2007. It was a very small sample size study, but yet the data have borne out, and it has to do with strokes. And there were people who were prior smokers, and they all had strokes. They were studied at the University of Iowa, where they had a large patient pool with different lesions. And individuals who had strokes that did not involve the insula didn't produce very much in terms of quitting smoking. So here, out of 69 total individuals, some of them had insula strokes, some of them had non-insula strokes. And this red area refers to the people who quit and stayed quit, and it was a large fraction of those who had strokes in the insula, more so on the right than on the left. Now, you can see it's a very small sample size. It was only 5 people who quit out of 6, but it was 5 out of 6 who had right insula strokes. So again, very small sample size, and this is essentially a natural lesion study, but it's been very well reproduced in further studies. So then the other area that shows a deficit in individuals who use drugs other than alcohol is the orbitofrontal cortex. And the orbitofrontal cortex here, the medial orbitofrontal cortex shown in red, is, again, it's very well connected throughout the brain. It's connected to the thalamus, so it gets information on every sensory modality. It's connected, it's not shown here, but it's connected to the hippocampus so that it gets data that helps put the information from the sensory data in the context of experience and a person's history. So it's very involved in decision making and linked to striatum in terms of its ability to influence an organism's response to seeing reward or other stimuli that are in the environment for which the person or animal needs to make a decision. So there are structural differences in the brains of people that we just saw from the Enigma study. But there is a study that came out from the Cambridge group in 2012 comparing people who use stimulants and their non-addicted siblings with healthy controls who don't use stimulants. And what they found was two things: the stimulant users and their siblings both had deficits in insula volume compared to healthy controls. And they also had larger gray matter volume in the putamen and the amygdala. And because of this study of siblings showing the same effect as their brothers and sisters who used stimulants, the conclusion of that study was that there's a vulnerability factor that leads to psychostimulant use. But whether or not the brain difference preceded the use of psychostimulants certainly was not proven there. And there are other studies that are more recent, that have recently been doing longitudinal analysis of people before they started taking the drugs. I mean, how do you know whether a difference in the brain is due to the drug or whether it was there before? One way is to do animal studies, which are not always completely translatable to the human studies, where you know very well what the behavioral state is, and you know very well what the drug history of the animal is, so you can control it. The other way is to study kids. Start studying kids before they take drugs and then study them long term. And the first of these studies was one that was of alcohol only, and a very, very nice paper came out in 2018 that gave the results of that study. And what they showed is that they started adolescents before they were drinking, starting at 12 years old, and studied them for throughout the adolescent development period. And what they were showing was differences in gray matter volume, differences in individuals who were heavy drinkers as compared with low drinkers, and looking over the decline over time. And the areas that are here in orange show the largest decline. They include the posterior cingulate cortex, which is important in memory and also decision making. Big differences in the superior frontal gyrus and the cingulate area. So this is the new kind of study. It's the longitudinal study of changes in brain development over time. Now, of course, you're really not able to control everything in the research participant, but you do get a good idea of when they start drinking. And of course, when they start drinking, are they also smoking? Are they also using marijuana? So it's very difficult to get a large enough sample to control for all of these variables. And I would say the largest long-term US study of brain development was started recently, maybe 6 years ago. It's called the ABCD Longitudinal Study. There are 21 sites. One of them is at UCLA. Another one is, they have them all over the country. And the goal is to study children from the ages of 9 to 10 for a 10-year period. They get structural MRI, the same functional MRI tasks, all of these different centers, and there's a coordinating center that is harmonizing the data, and they're just beginning to come out. So the idea is, why are you doing these imaging studies, right? The whole goal, it's not just to understand what's different about the brain of somebody who has an addiction, but there are targets that we'd like to fix. They're behavioral targets: craving, negative affect, withdrawal, problems with inhibitory control, deficits in decision making. These are things that we believe perpetuate addiction. And in order to target them appropriately, either with medications or now we can do it with brain stimulation techniques, we need to have a good understanding of what the abnormalities are. We now know that there are corticostriatal and corticolimbic differences in individuals who use drugs of abuse. We know that from the brain imaging studies. We know that there are certain biochemical targets. Dopamine receptors have been studied quite a bit because the first positron emission and SPECT radio tracers that were available were for the dopamine receptors. So there's an awful lot on D2 receptors, dopamine transporters, and now people are beginning to look at other biochemical targets that can be addressed with medications. So the first question you might have is, how does someone get a PET scan done? What is a PET scan? And what happens is that the patient or the research participant receives an injection of a radioactive tracer, which is a molecule that's of biochemical interest. It's taken up into the brain according to the biochemical behavior of that molecule. And the person is in a gantry, which really has a collection of detectors that will detect the decay of these radio tracers. And it's called positron emission tomography because the isotopes that are used, carbon-11, fluorine-18, nitrogen-13, they decay by positron emission. And there are 2 photons that are emitted in coincidence, and the detectors are connected to a computer system, which is going to reconstruct an image of where that radioactivity accumulated in the brain. So the tracers can be tracers for brain function, like radio-labeled glucose analogs, or they can be radio tracers that bind to specific neurotransmitter receptors or transporters. And one of the studies that you might find of interest is the study of cocaine craving. The first report came out in 1996. And the way the study was done was we had videotapes that the research participants viewed while fluorodeoxyglucose, a radio tracer for glucose metabolism, which is an index of brain function, was taken up into the brain. So there were videotapes that were very evocative. It made the people say that they craved cocaine like mad. And there were control tapes that were tapes of people doing arts and crafts, which did not induce cocaine craving. And these glucose metabolic maps show differences between individuals. And there were people who were low cravers and there were people who were high cravers. And here you have the picture of glucose metabolism in the brain when a person was viewing the neutral cues, the arts and crafts, and the cocaine related cues, which was, you know, white powder, money, people passing around a crack pipe. And for the low craver, there's really not much difference between the neutral and the cocaine related cues. But for the high craver, there's a huge difference. And there was a tremendous increase in glucose metabolism in the dorsolateral prefrontal cortex, as well as the medial temporal lobe, including the hippocampus, parahippocampal gyrus, and amygdala. And the degree of craving was correlated with the change in brain function in those areas. So here's the high correlation between the change in craving from neutral to active condition and glucose metabolism in the medial temporal lobe and in the dorsolateral prefrontal cortex. The other thing that we know, other than the brain function measured with glucose metabolism being correlated with craving, is that when somebody has craving induced by these cues, there's dopamine release, and there's a correlation of dopamine release with the degree of craving. So here, carbon 11 raclopride, which is a radio tracer that binds to the D2 dopamine receptors. And here's a beautiful picture of the binding of raclopride in the caudate and the putamen. And that is a PET scan in the neutral condition. But you can see that there is a correlation of craving with how much dopamine release there is. And this area here in red in the putamen is the area where the amount of dopamine release was highly correlated with the self-report of craving. Very strong relationship. And dopamine release was measured by using these raclopride scans to evaluate raclopride binding in the neutral condition where there's no cocaine-related cues and in the cocaine-related condition. And what you see is there's a change in binding across conditions. And this change in binding reflects the competition of dopamine that's being released with the radio tracer. So there is a decrease in tracer binding when people are viewing these tapes that are inducing craving. So dopamine is released, and the degree of dopamine release is related to how much craving somebody will report. So craving involves altered function in the cortex, the medial temporal lobe, and change in dopamine release in the striatal. So craving is one of our behavioral targets. Negative affect is another one of our targets. And here in a study of individuals who had methamphetamine use disorder, they reported their own evaluation of their depression. And it's tremendously different. I mean, 18 is a score on the Beck Depression Inventory that's so high that when somebody tells us that that their score is 18, we call a therapist to evaluate them for suicidality. They also score high on anxiety, and these scores on the Beck Depression Inventory and also the anxiety scale are correlated with their self-report of how much methamphetamine they use per week. And these are scores that are obtained when people are off the drug. They've been in the clinic abstinent for at least 4 days. Also, the depression and anxiety are correlated with brain function. And these are maps showing the correlation of depression with glucose metabolism measured using positron emission tomography. And you see that there's this very high correlation between metabolism in the amygdala and depression, as well as correlation with anxiety as well. These are positive correlations. On the other hand, there's a negative correlation between anxiety and activity in the prefrontal cortex. So you have, in addition, the indication that there's kind of a loss of cortical control of feeling state. We know that prefrontal cortex is connected to amygdala, and cortical activity can in fact reduce response to anxiolytic situations with respect to activation of the amygdala. So we have craving, we have affect, we also have impulsivity as a target, a behavioral target that we think is important in addiction. And one of my colleagues, Steve Reese, has taken the broad impulsiveness scale and created a biphasic model, which is actually psychometrically more correct than the triphasic model that's been published. And here we see the scores on the broad impulsiveness scale for individuals who are, they have methamphetamine use disorder, as opposed to individuals who do not use drugs, and whether it's the cognitive scale of impulsivity, which relates to planning, or the behavioral scale of impulsivity with items such as I have trouble keeping still. Individuals who have stimulant use disorder see themselves as more impulsive than individuals who don't use drugs of abuse. And this impulsivity is related to dopamine D2 type receptor binding. And here we've got plotted, the color is from a group of PET scans average for methamphetamine use disorder and for individuals with healthy controls. And really what we're looking at here in color is the statistical significance of the correlation between impulsivity and receptor binding. And here in the healthy controls, you see really just kind of a ribbon within the lateral putamen where D2 receptor availability is correlated with impulsivity. It's a negative correlation. Those individuals who have fewer binding sites are more impulsive. You see a much more robust relationship in individuals who have stimulant use disorder. This is throughout the caudate and putamen, and we believe that this relationship is much more robust than in healthy controls, because the healthy controls have what we refer to as spare receptors. The individuals who have stimulant use disorder have a 14 to 15% reduction in receptor binding, which really makes it so that perhaps each receptor counts more for them. Individuals with stimulant use disorder are not alone in the drug abuse field with respect to having a deficit in D2 dopamine receptors. This is work that was published by the director of NIDA from the Brookhaven National Lab, and she showed that individuals with cocaine, methamphetamine, alcohol, or opiate use disorder, they all have low binding of D2 dopamine receptors throughout the striatum. This is true for tobacco use disorder as well. And in fact, there's a sex difference so that men, but not women, who have tobacco use disorder show this deficit. Actually, that work was done by Dr. Amira Brown, who I believe is still faculty at Drew. She did that while she was with us at UCLA. Now, having a deficit in D2 type receptors is something that we believe is very important with respect to impulsivity and decision making. And this follows from the work of Logan and Cowan from a long time ago, where they defined roles of D1 and D2 receptors within the striatum. The D1 receptors are low affinity, which means that they're not activated unless there is a lot of dopamine in the medium or in the extracellular space. And so they are activated when there's a burst of dopamine release. And these D1 driven systems initiate the direct pathway or the GO pathway, which interacts directly through the globus pallidus to reduce activation of the, reduce inhibition of the thalamus. So the thalamus can drive activity to the cortex and increase corticospinal pathway activity. And the thinking is that activating this D1 system or this direct pathway allows an organism to know that it's time to react. And there are a series of possible responses. And the organism, if the organism is impulsive, will not take the time to select the appropriate response. The D2 system is involved in that kind of modulation and inhibition of the impulsive response. D2 receptors are high affinity. They're activated by the lower concentrations of dopamine that are available tonically, and they initiate activation of the indirect pathway or the no-go pathway. Indirect because it gets to the globus pallidus pars interna indirectly through the subthalamic nucleus, ultimately inhibiting activity of the thalamus. And we believe that the indirect pathway is important for an organism in terms of selecting which of the possible responses he or she can make is the appropriate response. Now, there's an imbalance between the D1 and the D2 system. And here you see PET scans of D1 type receptors and D2 type receptors in the striatum of healthy control individuals and individuals with methamphetamine use disorder. You can see that there's no difference in the D1 between the controls and the individuals with the drug use disorder. Each of these dots represents data from one person, and the lines give you where the mean would be for the group. There's no difference in D1. D2, on the other hand, you see visually, you can see there is a deficit, and it's essentially a 15% deficit. Again, each dot is a person, and there is less D2 receptor binding in the individuals with methamphetamine use disorder. This is true for cocaine use disorder as well. There's no difference in D1, but a deficit in D2. And we think that this would produce an imbalance between the indirect and direct pathways and would contribute to the impulsivity. So aside from impulsivity, which is essentially a personality trait that's measured from self-reports where people tell you how they think they behave in the world, we can also measure decision-making in the laboratory with objective tests where people make decisions and we measure their responses. You can also do this while people are having their brain activity evaluated using fMRI. So in the real world, the kinds of decisions that somebody has to make are between drug and sobriety, criminal versus legal activity, risky versus safe sex. In the laboratory, we can use largely monetary tasks where we can evaluate how someone can devalue a reward on the basis of whether or not it's in the future, whether they want something immediate or can take a larger reward later on. There are gambling tasks where individuals who are impulsive will primarily go for high gain, even though there's higher loss. And then we also have a task that we've used in our laboratory, which is a task of uncertainty under risk, the balloon analog risk task. And so we can do these, as I said, with functional magnetic resonance imaging, and MRI, as we already mentioned, has to do with measuring activity when a radio frequency pulse that was applied is removed and there's a signal that can be detected by a coil around the brain. And the technique that's used for fMRI, or functional magnetic resonance imaging, is called the BOLD technique, which is the blood oxygen level technique. And what you can do is you can measure the difference in the oxygenation of blood. What happens when you go from the basal state to the activated state? You have increased blood flow, which is going to bring more oxygen to the tissue that's active. But what happens is there is more oxygen delivered than is actually utilized. And so you have a change in the ratio of oxy to deoxyhemoglobin, and that'll produce the bold signal. So you can see bold signal reflecting increases in activity in different regions of the brain. And we can do that while people are taking the balloon analog risk task. And you can imagine what this task is all about. There is a screen that people can view while they're in the scanner. And basically, every time they're looking at the screen, they have a balloon that shows up. And the balloon can be increased in size if the person makes the decision to pump it up. And every pump makes the balloon a little bit bigger and increases the value of the potential reward by a quarter. But the balloons are programmed to burst after a certain number of pumps. And so what happens is each pump carries potentially greater reward, but there's also greater risk of the balloon bursting. And what you can do is you can measure the degree to which the amount of risk or reward will modulate brain activity. So the question that you're asking is, to what extent is brain activation influenced by how much risk somebody is taking? And in healthy people, what we see is that activation in two regions of the brain, the insula, which we talked about is important in decision making, and the dorsolateral prefrontal cortex, these regions of the brain show a dependence on the number of pumps that someone is taking. So these regions show an activation that is dependent upon the level of risk. And by the way, that level of activation is correlated with dopamine receptor availability measured with positron emission tomography. So the same people were tested with fMRI and then with PET, and those who had the greatest number of dopamine receptors showed the least modulation by risk. And these are healthy controls. We think it's quite different in individuals who have stimulant use disorder. And the activation in the ventral striatum when they decide to cash out is correlated with the amount of reward that they're going to receive. And here again, the bold signal in the striatum is correlated with dopamine receptor availability in the striatum as well. So dopamine receptors in the striatum are important in the degree of activation of the cortex or the striatum when someone is deciding to actually take the risk or deciding to cash out and avoid the risk. There's a big difference between healthy people and methamphetamine users in that while the dorsolateral prefrontal cortex is important in the modulation by risk in healthy controls, individuals with methamphetamine use disorder have much less of the bold signal in the cortex dependent on how much risk there is. In addition, what they find, which is kind of an opposite effect, is that the individual with stimulant use disorder, when they're deciding to take the risk, show a modulation of activity in the ventral striatum, unlike the healthy controls. So the brain of the individual with stimulant use disorder differs from that of the brain of the individual who is a healthy control with respect to how risk is considered. So all of this because of the observations that we've made that D2 dopamine receptors are involved in addiction suggests that what we'd really like to do is upregulate those dopamine receptors so that people would be less impulsive, make better decisions, and have just a different trajectory in treatment. And so based on animal studies where exercise increased the viability of D2 type dopamine receptors, based on rodent studies, we did a study of exercise as an adjunct to treatment as usual for individuals with stimulant use disorder. There were two conditions. There was a condition where they had 8 weeks of exercise training involving aerobic and weight training for 40 to 50 minutes. And every single person in the exercise group showed an upregulation of the striatal D2 dopamine receptors to the level of controls. The control condition was an equal amount of time devoted to a health education interactive session, and there was no change in the D2 dopamine receptors. So, of course, we're evaluating now whether this increase in the D2 dopamine receptors is associated with improved abstinence in individuals who are in a treatment program. Of course, it's not all about dopamine. There was an awful lot done with dopamine way back when, but now we can study other targets. And so there's a lot of interest in microglia, inflammation, transporters, different glutamate receptors. And so I can tell you that inflammation was considered a major target because there was a study by a Japanese group showing that there was marked inflammation in the brain of individuals with methamphetamine use disorder, measured with positron emission tomography and binding to the transporter, the mitochondrial transporter, which is elevated in microglial activation. We did a study and we found that there appears to be essentially a 10% difference between individuals with methamphetamine use disorder and healthy controls. It was not statistically significant, and in fact, treatment with abutolast in this population didn't seem to work. Other targets, glutamate. FPEB is a ligand that binds to the mGlu5 receptor, which animal studies tell us produce vulnerability with respect to animals with high binding in the striatum, taking a lot of cocaine or methamphetamine. But the mGlu5 receptor is also important in cognition. And here in a very preliminary look, data that we generated before COVID shut us down, you see a dramatic deficit in individuals with methamphetamine use disorder throughout the cortex as compared with healthy controls.
Edie,
So,
can you do about 5 more minutes? Oh, you're even done. Okay, even better. Thank you.
I'm done. I just
Okay,
want to
perfect.
I just
Perfect.
want to thank my colleagues and thank you, Theodore, for trying to keep me on time. But I rehearsed. I tried to keep it on time anyway.
Perfect.
We have wonderful colleagues at UCLA. I've had a wonderful group of trainees. Maybe some of the people from Drew can come into our T32 program. We want pre-doctoral or postdoctoral candidates. Keep it in mind, and we'll try to move your master's level candidates along. And thank you for the support from NIDA and Foundation. That's it.
Great job. Very nice. Thank you, Edie. Time for questions. I'll ask the first question.
Sure.
Is impulsivity and risk-taking synonymous?
No.
Okay. Can you explain the difference?
Well, you know, impulsivity is a broad construct that has subconstructs. So, for example, impulsivity generally often can mean acting without thinking. Risk-taking is a different kind of thing. Individuals, some people prefer risk.
Mhm.
They are risk-seeking. There is an association of risk-seeking with sensation-seeking. All of these things probably are best broken down separately and considered separately. And they also likely have different neuroanatomical and possibly neurochemical substrates.
Okay, great.
Dr. Landon, I have a question too. In the beginning, when you showed the brain scan studies, I think the longitudinal one about adolescents and the structural changes in those who were drinking more heavily, are those reversible?
Well, those longitudinal studies haven't been extended throughout time, but there's something important for you to be thinking about. And that has to do with when the drug use occurred. So think about this. The brain continues to grow and develop through the age of 35. And then for people like me, it's all downhill. But what happens is gray
And
matter.
the 4 years since you've been 35.
Right.
Right.
Thank you, Theodore. The gray matter is pruned, and so that circuitry becomes more defined. When children or adolescents are in fact drinking or using drugs of abuse during that period of time, they are altering perhaps the trajectory of brain development. Brain development is happening there, and whatever happens then could be a forever sort of thing. The other thing that's happening is white matter pathways are evolving. And so the connections, especially between the prefrontal cortex and the striatum, that are so important for inhibitory control, they're being developed at that time as well. So of course, if an individual stops using, there's going to be some recovery. I can tell you that we just published a paper like last month on a study that could never have been done in the United States. It was done in China, where people who were caught with a certain amount of methamphetamine on the street are sent to compulsory treatment. And these people were in compulsory treatment for up to 2.5 years. And so we got their brain scans from early abstinence all the way through later on. Now, if you were to take a group of people who were abstinent for varying periods of time, compare them with age-matched controls, there's no significant difference. But if you take a look at the individuals who use the methamphetamine, early in abstinence, what you see is you see a deficit in striatum and a bunch of cortical areas. And then in the individuals who are abstinent for 2.5 years, you see larger volumes, larger gray matter volumes. So there are changes, there are adaptations that occur. The extent to which these adaptations are functional is not known. So I can tell you that with cognitive deficit, which methamphetamine users definitely have, there is an improvement in the overall score across a bunch of different cognitive tests in just a month. There's an improvement. With the D2 dopamine receptor deficit, the only improvement that's ever been reported is in that exercise study that we published in 2015. Dopamine transporter, which I didn't really talk about because I wanted to stay within the time limit, does show improvement in the thalamus, but not the striatum, over 2.5 years. 2.5 years.
Oh.
So there is improvement, but you know, in terms of clinical dealings with clients and patients, it's important to stress that patience is important.
Yeah. And then what did you mean by compulsory treatment that the paper in China was doing?
Well, it sounds like just like what I said. They weren't asked, would you like treatment or would you like not to have treatment? They were
Oh,
told you are going into lockdown where you will have treatment. And
wow.
they were not allowed to have any drugs of abuse. They were not allowed to have visitors. They were allowed to smoke no more than 1 cigarette a day. Now, I don't know what the rest of the treatment was like, behavioral therapy.
Yeah. Okay.
So it's kind of a different approach to treatment than what we
Yeah.
have.
Slightly. Yeah. Okay. Thank you.
My pleasure.
Yeah. Hey, Edie. I was just thinking that those Chinese studies in like early 2000s where they were lesioning the nucleus accumbens, right?
Yeah. That was, you think that was compulsory treatment?
I think that, well, I think they actually, you know, they were probably given options, but that was one of the options, was to go for surgery and have your nucleus accumbens lesioned, electrically lesioned with a little.
Did it help?
I'm not sure.
No.
Given option.
There was a lot of depression afterwards, but it's published in some of the surgery journals.
Wow.
Yeah.
So in terms of trying to get a paper published, when the data were collected in China, the referees ask a lot of questions about informed consent, as they should.
Yeah. So did you have to get the IRB through UCLA? Did you?
Of course.
Yeah, that must have been complicated as well.
Well, I didn't have to get permission to provide compulsory treatment. I just had to get permission to
Use
analyze
the data. Yeah. Yeah.
Okay, trainees, do we have any questions for Dr. London or Dr. Evans? Dr. Lutfi's on, Dr. Shao's on. Dr. Sanahekam's on. Do we have any questions or do you want to discuss any kind of projects you might guys have? Delara, do you have any comments or questions? Anna?
I do not have any questions, sorry.
Okay.
Oh, here, I will stop the recording actually.